The overall aim of this experiment is to achieve high transfection efficiency in human-induced pluripotent stem cells using optimized transfection and nucleofection protocols. This is accomplished by first establishing feeder-free cultures of human-induced pluripotent stem cells. For transfection, the human-induced pluripotent stem cells are plated on Geltrex or Matrigel overnight, while for nucleofection, small cell clumps are obtained by trypsinization. A plasmid DNA and gene-induced transfection reagent mixture is added to the plated cells, and then the plate is spun and incubated at 37 degrees Celsius. The human iPS cell suspension is nucleofected in a nucleofector device, and cells are resuspended in MEF-conditioned medium and incubated at 37 degrees Celsius. Ultimately, results can be obtained that show efficient genetic manipulation through high transfection efficiency in human iPS cells.
The main advantage of this technique over existing methods like previously described transfection, electroporation, and nucleofection methods is that high transfection efficiency in human iPS cells with consistent outcomes can be routinely achieved in our lab.
Our protocols also ensure minimal cell death following genetic modification.
Hence, our protocols permit a rapid and large-scale generation of transgenic human iPS cell lines.
Prior to adapting human-induced pluripotent stem cells to feeder-free cultures, prepare Geltrex-coated dishes. Next, passage the human hiPSCs previously maintained on feeder cells. Add 1 ml of acutase per well and incubate at 37 degrees Celsius for 1 minute until most cells start to detach. Add 10 to 15 glass beads to the cells and gently swirl the plate. Then add 2 ml of knockout DMEM F12 and gently triturate. Transfer the cell suspension to a 10 ml centrifuge tube. Spin the cells at 800 rpm for 3 minutes at room temperature. Aspirate the supernatant, leaving the human iPSC pellet intact. Gently flick the tube to disperse the cell pellet. Gently resuspend the human iPSC pellet in an appropriate volume of stem-prone medium. Distribute between the wells of a Geltrex-coated plate. Depending on the proliferation rate, human iPSCs can be passaged in a split ratio of 1 to 2 to 1 to 6. Carefully place the plate into a 5% carbon dioxide 37 degrees Celsius incubator and swirl the plate gently to ensure an even distribution of cells across the wells. Feed cells daily. When cells reach 80% confluency, they're ready to be passaged again onto a new Geltrex-coated well at a split ratio of 1 to 2. Cells should be approximately 40 to 50% confluent and in small colonies distributed evenly on the Geltrex-coated well on the day of transfection to achieve optimal transfection efficiency. To begin this procedure, prepare 100 microliters of knockout medium in a sterile 1.5 ml Eppendorf tube. Add 27 microliters of gene-induced transfection reagent. Mix well and incubate at room temperature for 5 minutes. Next, add 4 micrograms of plasmid DNA. The choice of plasmid is critical for optimal transfection efficiency. The plasmid used here has an enhanced green fluorescent protein driven by a CAG promoter, which is a strong promoter that is transcriptionally active in human iPSCs. Incubate the tube at room temperature for 15 minutes. After 15 minutes, add the gene-induced DNA mixture to the cells and swirl the plate. Spin the plate at 1200 rpm for 5 minutes to increase the contact of transfection mixture with human iPSCs in the well. Incubate the cells at 37 degrees Celsius overnight. On the following day, monitor for transfection efficiency. On the day of nucleofection, Pre-treat human hiPSCs with 10 micromolar ROC inhibitor for at least 1 hour at 37 degrees Celsius. While the human hiPSCs are being pre-treated, prepare 82 microliters of human stem cell nucleofector solution in a sterile 1.5 ml Eppendorf tube. Add 18 microliters supplement 1 and mix well by pipetting. Incubate the solution at 37 degrees Celsius for 5 minutes. Pre-warm previously prepared feeder cell conditioned media and 0.25% trypsin EDTA to 37 degrees Celsius. Under the hood, pre-label one sterile 15 ml conical tube. Carefully remove the media from the human hiPSC culture to be nucleofected and gently wash the cells with phosphate buffered saline. Aspirate the PBS and add trypsin. Incubate cells at 37 degrees Celsius for 3 minutes.
For successful nucleofection, it is critical not to trypsinize the cells into single cells. Instead, the cells should be triturated into small clumps consisting of 2 to 3 cells.
Gently triturate the cells with a P1000 pipette tip into small clumps consisting of 2 to 3 cells. Wash the bottom of the well by repeated pipetting to make sure all human hiPSCs are completely detached. Then transfer the cell suspension to the labelled 15 ml conical tube. Next, add 9 ml of mouse embryonic fibroblast media to inactivate the trypsin. Spin the cells at 800 RPM for 3 minutes. Carefully aspirate the supernatant, leaving the cell pellet intact. Resuspend the cells in 100 microliters of pre-warmed human stem cell nucleofector solution. Using a 1 ml pipette tip, transfer cells to a nucleofector cuvette. Add 4 micrograms of plasmid DNA into the cell suspension in the cuvette. Mix cells and DNA by gentle swirling. Tap the cuvette twice on the hood surface. Insert the cuvette into the nucleofector holder of the nucleofector device. Select program B016. Nucleofect cells by pressing button X. OK will be displayed when the nucleofection process is completed, which usually takes 1 to 5 seconds. Use the plastic Pasteur pipette provided in the nucleofection kit to retrieve nucleofected cells from the cuvette. Recover cells by resuspending them in pre-warmed CM and ROC inhibitor in a sterile 1.5 ml Eppendorf. Incubate cells at 37 degrees Celsius for 10 minutes. After 10 minutes, use a 1 ml pipette tip to transfer the cells dropwise onto feeder layers. Incubate the cells at 37 degrees Celsius overnight. On the following day, monitor for transfection efficiency. Shown here are photomicrographs of EGFP-expressing RIV1 human hiPSC cells transfected using gene juice on Geltrex 12 hours post-transfection. Plasmid DNA transfection of human hiPSCs using gene juice with a strong promoter typically yields more than 60% of EGFP reporter-expressing cells in a transient transfection assay. EGFP-expressing cells can also be obtained by nucleofection, as illustrated in these images of RIV1 human hiPSCs plated and formed on feeders following nucleofection using either the B016 nucleofection program or the A023 nucleofection program. The use of the B016 program results in up to 80% of EGFP-positive cells, but the A023 program yielded less than 10% of EGFP-positive cells. Furthermore, stable clones of transgenic human hiPSCs that express the transgene can be derived. An example of stable EGFP-expressing human hiPSC colonies with ubiquitous EGFP expression derived from gene juice is shown here. These cells retain constitutive EGFP expression during embryoid body differentiation.
After watching this video, you should have a good understanding of how to nucleofect and transfect human iPS cells.
Efficient genetic manipulation is an essential tool to study various aspects of stem cell biology
and will accelerate the use of human iPS cell lines in regenerative medicine.
The advantages. The protocols result in simple, robust, and highly reproducible techniques to introduce transgenes into human iPSCs without prominent toxic effect and cell death. Transient transfection assay generates large numbers of transgene-expressing human iPSCs within 1 day. Stably transfected iPSC clones usually appear within 7 days, and these transgenic colonies should be ready to be picked within 3 weeks. The protocols can be fed into other applications, including overexpression, conditional induction, derivation of lineage-specific reporter lines, shRNA or siRNA knockdown, gene targeting and homologous recombination. Take into consideration. Human iPSCs should be passaged into smaller clumps of cells, 5 to 10 cells, and plated on gel tracks at high density, 1:2, to ensure optimal transfection efficiency in numerous small colonies. Transient transfection assay generates large numbers of transgene-expressing human iPSCs within 1 day. For human iPSC lines that are more prone to differentiation and cell death, higher number of human iPSCs, up to 4/106 cells, should be used for a single nucleofection experiment. Thank you for watching. Subscribe if it is useful.